In this video, we're going to talk about genomic libraries. Now, in the previous video, I talked about vectors and how to put your DNA sequence into that vector. So the natural question is, where do we get a DNA sequence from? Let's say we want to study protein X. Where do we get the DNA sequence for protein X? Well, we can get them through a genomic library. And a genomic library is basically the clone set of every single DNA fragment from a particular organism. So the genomic libraries exist for bacteria, yeast, mouse, human, flies, and a bunch of organisms. And they're owned by companies, so you can buy those for your research purposes. Now, the way to make a DNA genomic library is by what is called shotgun cloning. And basically, what you do is you take the chromosomal DNA of the organism you're interested in, you cleave it to clonable sizes. So remember, in the vector, we're looking for sizes less than 10K. And then you put that into a cloning vector. Now, there's some math to go with the shotgun approach. And it's all based on probability. So how many randomly generate 10 kb sequences? Because that's kind of what you're doing. You're taking the chromosome and more or less randomly breaking it apart. How many sequences must you make to make sure that 99% of all DNA, or all, E. coli sequences are in your library? Now, that is given by these two equations, where probability of coverage, right, equals 1 minus 1 minus your fragment length. So here it's 10K raised to the power of n. N's our number of fragments. So we're actually trying to figure out n. And by doing some math, or algebra rather, we can have an equation for n alone. So our number of fragments equals log of 1 minus probability divided by log of 1 minus f. And I made a little bit of an error. F is actually desired fragment length divided by total genome. So here is 10,000, or it's 10 kilobases divided by 4,639 kilobases. That is your size of your E. coli genome. And if we solve for n, with probability being 0.99, we see that if we create 2,134 random fragments, we are 99% sure that we have any protein that we want. Now, that's one way to get sequences, the shotgun approach. Another way is through what's called a cDNA library. Now, the way a cDNA library works is that first you isolate all of the mRNA from a cell. And if you remember mRNA, when DNA is going to make a protein, the first thing you do is you make an RNA copy. If you're in eukaryotes, you remove the introns. In prokaryotes, you don't have introns, and you get mRNA. And it's this mRNA that is being read in to make proteins. Well, in a cDNA library, what you do is you purify these mRNAs, and you use what's called a reverse transcriptase, which is an enzyme that reads in RNA and makes DNA from it. So what you're doing is you're isolating all the mRNA and just turn it into its DNA form. So you're reverse transcripting it. We call that complementary DNA, or cDNA. And with this technology, we can create what's called a microarray or DNA chip. So the way a DNA chip or microarray works is that we take our cDNA and we put every single copy of our cDNA on a well on this chip. And the reason you do that is you want to check mRNA levels inside of a cell, for an example. Let's say you're looking at a cancer cell versus a non-cancer cell, and you want to see in your cancer cell which proteins are being overexpressed or underexpressed. You can do that using a DNA microarray. So you take your non-cancer cell and you just measure your mRNA levels. It tells you what proteins are being made. Compare that with your cancer cells. And you can dye these different colors, right? Your control cell can be green, and your cancer cell can be red, and your readout looks like this. If you get yellow, nothing bound. If you got green, your control bound. If you got red, then your cancer cell is producing more. And you can also look at what's called the exome, which is what mRNA is in this cell at that given time point. Now let's say you've made a cDNA library and you want to get a specific gene out of that. How can you tell which bacteria has which gene? So here we have bacteria with different vectors on them. And what you can do is you can transfer these bacteria to a filter. And on this filter, you are going to let that filter dry, and then you're going to wash over your single-stranded DNA to see which bacteria has your vectors. So let's say, again, we're looking at protein X, right? So you want to wash your filter with a probe that will be able to detect protein X. So your DNA of interest will bind to your labeled probe. And then you're going to measure this binding event. So your probe usually has fluorescence, or it could have radioactivity with it. And only the colonies that had your DNA sequence will come back as having a hit. So if you know which colonies had your DNA sequence, you can go back to your master plate and you can pick out those colonies to grow your vector in. And so that's how we not only create a DNA library or cDNA library, but this is how we actually determine which bacteria have our sequence of interest that we can use to go and put into our vector and start producing our protein. And that is it for this video, and I will see you in the next video.